Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's Neuroscience Research Podcast. Today is March 20th, 2025, and it's the day of our annual Neuroscience Symposium. We've listened to the symposium over the course of the day, and this will be our panel discussion. The symposium was entitled Prefrontal Cortical Functions Beyond Fear. And I didn't make this title, but if I could try to sort of explain the background of the title, I would try. And Tony made up the title, maybe if I get it wrong. But I'm trying to explain what it means, and I would say that the prefrontal cortex is known to be deeply involved in learning a particular kind of task, which is conditioned fear task. And this is measured by preceding a shock with a neutral stimulus, like a light or a tone, and without any previous experience, a rodent will quit moving when the shock comes, which is called freezing, but not to the neutral stimulus. And if it gets repeated enough times, the animal will start freezing to the neutral stimulus. And then if you take the shock away and keep giving the neutral stimulus, the response to the freezing response will extinguish. And so that's a kind of set of phenomena that get studied a lot, and it's pretty easy to ask what happens if I inactivate the prefrontal cortex or some other cortical region or some other subcortical region. And a lot of that has been done, and it's been very good. It's all been very fruitful. Mm-hmm. And we have actually discussed it on the podcast quite a bit before. That's right. I think the idea today is to go beyond that. The cortical regions are complicated. They don't just do one thing. It's not likely that the prefrontal cortex's only job is to control freezing in response to shocks. And so how can we build on what we know about prefrontal cortex and fear conditioning to develop a more expansive view of what the prefrontal cortex's job is in learning or in other things? So if I could get away with that description of what it was about, we have 4 different speakers who took quite a bit different angle at it, which is expected because the whole goal is to branch out from what's been done before. And so we expect different branches that are different. So maybe I'll start by just having everybody introduce yourself, say where you're from. Thank you for coming by. Tony?
Oh, I'm Tony Burgos, and basically I'm an assistant professor here since 2019 at UTSA, and I'm originally from Puerto Rico, but something brought me to Texas. I've been really happy to be here, joining this session and helping to organize it, especially bringing experts from the field to talk more about prefrontal function.
Tony organized the symposium, and if you follow the podcast, you know him well. Anyway.
I hope so.
I'm Laura Denardo. I'm also an assistant professor, but at UCLA, and I started my lab in 2019 and got interested in PFC and its role in fear, learning, and memory, actually from reading a lot of Tony's papers, which I cite many times, have cited over the years, and now have become more interested in sort of time-dependent processes there.
I'm Jason Keller. I'm a research scientist in Josh Dedman's lab at the Howard Hughes Medical Institute, Janelia Research Campus in Virginia. I'm originally from Texas, though. Good to be back. And I'm broadly interested in how learned and innate behaviors interact, and kind of got into motor, cortical, and prefrontal control in that regard.
And I'm David Morilak. I'm from just up the road at UT Health San Antonio, soon to be merged with UTSA. I've been here. I bring some grace and wisdom to the panel. I've been a professor for some time. My first faculty position was at UT Health in 1994. I'm a stress neurobiologist and a long path toward understanding the neural responses to stress, how stress causes pathology, and then looking for entry points to therapeutic intervention brought me to the medial prefrontal cortex. That seems to be a nexus of all of those things in the relationship of stress to brain to behavior.
And I'm your host, Charlie Wilson. So I'd like to just make a wild stab at describing 4 different views that I heard you say. And I know I may not get it exactly right. Feel free to please correct me as much as you want to. But I thought I heard Tony talking about sort of the actions that the choices one has, and what to do when confronted with a fearful situation or threat. And one of them is to freeze, which may or may not be helpful. I guess rodents have learned to do that and mess up in some situation. Or to run away, fight back, to seek safety. And these are a bunch of things that in the laboratory can be made available to the animal to do, and animals will choose those, and you can try to understand what they do. And it sounded like he was thinking of cortical regions as engaged in these choices, action selection kinds of choices, which seems like a reasonable function for the cortex, and the kind of thing the cortex has been accused of doing many times.
Well, Charlie, you are usually very humble in, not coming from or coming from a different field, and typically, you take, really good shots. Basically, during threatening fear situations, there's this initial response, which we call freezing, right? Basically, animals stop moving around just in preparation for something to happen, right? But this is typically because the animal is just taking a response in preparation for not only something to happen, but to make some kind of decision, right? The most appropriate response you would imagine during a threatening situation could be, running away, as you mentioned, right? But there's so many choices, right? It just depends on the specific situation. And in this case, like a freezing response is typically even triggered at unconscious states, right? Based on cue associations from the environment and based on experience. But that doesn't mean that's the end of the response, right? It's not an all or none or binary situation in which the animal freezes or don't freeze. But after the initial freezing response, the animal could just decide what to do, right? Depending on an action-based response, if that action leads to any benefit, like avoiding, escaping the incoming danger, then why not to keep using that response in the future?
So freezing might just be a pause, think about what to do.
Exactly.
Take a moment to decide what to do next.
Yeah, that's the way I see it, right? Yeah. Take a moment and see, or just explore, study, what are my options, right? If there are options to escape, well, why not to do those?
We can also take an evolutionary perspective on that, though. I mean, the medial prefrontal cortex, the prefrontal cortex in general, is part of an old cortex. It's limbic cortex. So it is on the edges of the cortex, which is not the most evolutionarily elaborated part of cortex. So it's doing something sort of fundamental. And I wonder about interpreting different behaviors in a species-specific way, because the way I take it, and I like the title of your symposium, because you've described an example of what we call executive function, which is really what's been ascribed to this part of cortex. Executive function is kind of metacognition. And it really is, we view it as strategy selection, or choosing from multiple options, and then learning from those options what worked previously. So if you're a rodent and you find your food by running around in a field and discovering what you bump into, and rodents do exhibit motor behavior as a part of motivated behavior. If you hear a screech from a hawk who has evolved to detect motion, your best survival response is to stop moving. Right. That may not be true of a bear faced with a challenge, or a primate or something. I think what the prefrontal cortex is doing is strategy selection and then learning what works best and reinforcing that. But beyond that, it learns what no longer works and then chooses something else. And so this switching and shifting between habitual behavior, which is something that has been learned and paid off, versus now exploratory or experimental motivated behavior, I think that covers it's then emotion agnostic, because it can do the same thing for positive reinforcement or for avoidance or for learning things that are absent.
So even in the general way that you put it, there's still lots of different things being learned.
Right.
Like one is, what is this sensory context I'm looking at? How much of a threat is it? And probably not every part of the brain is doing a threat evaluation, but some part of the brain is. Maybe that's prefrontal cortex is part of that, or maybe it's just part of the next step, which is what, given the threat, how should I
We're given a choice
next?
Right. Yeah.
So, do we know where in that multi-layered decision path the learning in prefrontal cortex is critical? It's not doing all those things.
Well,
I think it's doing a lot of them.
exactly.
Yeah.
Probably all of them.
Because Jason brought up this interesting point that we see sort of this encoding of like what I think is probably a behavioral context where you can see activity before a cue even turns on. And we've shown in other studies that you can actually predict what the animal's going to do from that activity. And so that suggests there's this layer of behavioral context. And then on top of that is overlaid information about specific cues. And I really think about it, it's an association cortex. So it allows flexibility by helping animals link cues, which may change based on the context they're in, or they themselves may change with appropriate behaviors or suppression of behaviors. We also see a lot of the inhibition in the prefrontal cortex, I think, is just as important as excitation for shutting off probably competing.
So for some of it, I wonder if you mean all of it. For example, in the situation you gave, there's a visual stimulus or a sound, which is a hawk. And at some point, this animal learned that sound goes with hawk, and that hawk is a threat. So that's a visual or an auditory object identification. And this, normally that's not what we think prefrontal cortex does, but do you think it could be?
I was just going to add like the multi-selectivity point as well, is that a lot of people have recorded. I think that's something that's somewhat unique about prefrontal cortex is the high dimensionality. It does tend to encode motor acts or actions and sensory stimuli, auditory, visual. It can respond to any of these things and kind of co-respond to all of them at once or in any combination. It's like it's got this really high-dimensional character, which I think that's somewhat unique with respect to other cortices.
Yeah, what might be the most salient stimulus toward altering activity in the prefrontal cortex is the effectiveness of either an association to a response or to an outcome of a behavior. We've seen the same thing, as have others, that we have this test where animals discriminate and they have to make a choice between two locations based on cues, and then they either get a reward or they don't. And what we have found in some early studies is that in the medial prefrontal cortex, we get a response that's activated just before they make the choice. Then, when they choose and they dig in the pot and they get a reward or they don't, depending on the outcome, the behavior of the region differs. So it's differentiating successful from unsuccessful responses given a context. In another part of prefrontal cortex, we've seen the timing of the response shift as the behavior becomes well learned. And so it's now, I think, moving from learning an appropriate response to reinforcing an ingrained effective response. And then when the surprise comes, I imagine we'll see a reshift again. Very similar to what we see in dopamine neurons that are predicting reward and then responding when differentially whether the reward that's expected comes or doesn't, because then that warrants a new behavioral strategy.
And which projects to
prefrontal cortex.
And I guess, like all these characteristics of the medial prefrontal cortical signaling keep adding up to this view of the prefrontal cortex as a higher order executive function system, right? Just because it's very rich in the diversity of cells that are populating the different layers. It's very rich in inputs, it's very rich in outputs. It connects to not only emotion-related downstream regions, but also connects to motor regions, as Jason was talking today. But it's also, not only staying with projections within the brain, but those projections could extend all the way, to midbrain areas and the brainstem, and to try to bypass even other kind of signals occurring in the rest of the brain. Right? I think we have been tapping into many of these functions little by little, right? And I mentioned to them multiple times, that the more I learn about the PFC, the more confused I become, because you could not come up with a single explanation of what its function is. Everything is pointing towards this multidimensionality function, which perhaps is highly dynamic, even if you have requirements for learning something, but then you transition into something else. Like, for example, in the case of David's work, like reversal learning, right? The PFC is not stubborn, quote unquote, to keep signaling the old association, but is flexible enough to switch into the encoding of the new rule. And you keep changing the rules, and the PFC basically will kick in to keep changing those rules or changing.
That's such a good point. Sometimes the function of a region is best illustrated when it goes wrong, and you see what is the consequence of dysfunction, right? Which is the perseveration, the inability to switch. No problem learning, but difficulty in changing learning,
right?
Or shifting responses based on changes in the environment.
But this is telling you that within this multidimension, signals from past experiences are being processed at the very same time that the new pieces of information are arriving, so that in a flexible manner, the new rules or the meanings based on the signals from the environment could be reversed or slightly modified, edited, just as needed. Right? Taking into account also the feedback based on the response, the behavioral response of the animal or the human, right? In that case, if the outcome or based on the behavioral response that was taken was a negative one, perhaps, it's better to even learn from that faulty behavior, right? So that in the future, you don't try it again. You try something new, perhaps.
I just thought of a great experiment, right? A mouse in a field, and you give it a hawk screeching stimulus, it will freeze. Fox on the ground, now a visual proximal stimulus, it's going to run away, right? Make the fox screech like a hawk and see what happens. Does it freeze or does it flee? And that would tell you something about, I'll bet your prefrontal cortex is going haywire.
Yeah.
You have to be recording for the prefrontal cortex.
And I think what you're saying taps into another core function of the PFC, which is to make predictions, right? Because you can't update so quickly if you're not making predictions. And there's been a lot of studies that have shown that predictions are encoded, especially in like ACC, for example. People have shown this. But I think it's making and updating predictions and using, cue outcome associations encoded probably in other brain regions to make a prediction. And then if, the outcome was bad, update quickly.
Exactly.
So that was exactly the kind of thing I was getting at, because if there is some function in this learning text that's performed by some other brain region, and the prefrontal cortex is relying on that, then we could say, what are the things that the prefrontal cortex doesn't do for itself, but relies on other parts of the brain to do?
Well, at least we know from Laura that at least very early in development, there's no really good PFC signaling during.
And one of the things that she said that was very positive about relying on other parts of the brain, is consolidation, the long-term memory, which seems to require the incorporation of information from other cortical areas. So at least in that process, that up to this moment, I've been thinking, you all are thinking, well, you don't need any more cortex except prefrontal cortex. It doesn't. You don't need a visual cortex to recognize a fox. You don't need an auditory cortex to recognize a hawk. You don't need any of that stuff. You just have a prefrontal cortex. Of course, it's not really what you're saying, I guess. So
yeah, I mean, what we're finding is that these cortical neurons become enriched in a trace of remote memory trace. And so we show that one pathway is important for remote memory, but I think probably there's a more general phenomenon going on here, which is that you're recruiting a set of neurons in PFC that can control multiple cortical regions at once and then you probably do need visual cortex and auditory and whatever to bring up sort of like a multi-sensory representation of an experience so that you can use all of that information to make a decision. And so I think that's one interesting thing direction that I'd like to go is to explore more what the collateral axons of these neurons, what those pathways are doing, and if there's some synergy that's important for bringing up that representation.
I was actually going to ask, I think that was your answer to the question I was going to ask, which or could be one answer, which is a question of to what extent prefrontal cortex is good to lump that all in one, because I know like we've done a lot of talking about prelimbic versus infralimbic or like these cortical connections. So I was just kind of curious to talk to people here about who've done a lot more work than I have about prelimbic versus infralimbic and all the differences about, to what extent these are playing different roles and have different anatomy versus lumping them. Can we really call them prefrontal cortex and try to come up with some?
Yeah, at least we know that there's some differences in connectivity, and you have to be into even older literature telling you a whole lot about inputs into the infralimbic cortex in contrast to the prelimbic cortex that are like more like visceral. So even like internal feelings are processed through the infralimbic cortex. That has perhaps to be processed within this multi-dimensional signaling and function. And I think like you tap into some of those aspects of that. Even single neurons can signal so many things, right? Perhaps adding even further into all this multi-processing function that characterizes even dorsal and ventral PFC areas. What are other differences? Well, at least we know that there seems to be more engagement of the dorsal PFC, like the prelimbic cingulate, when outcomes are bad, right, threatening, dangerous, versus when outcomes are more beneficial, like in David's work, right, extinguishing fear response. It seems, to give more of that cognitive flexibility, and that seems to engage more of the ventral PFC areas.
I think they're more complementary than opposing, and they do things differently in different contexts. But also, I think your question is a good one because it really highlights a shift in mentality in just neuroscience research in general. We're not asking what does this region do or what does this region do in concert with a target. We're talking about what do subpopulations of overlapping interdigitated circuits do. And there's a reason that they live in the same real estate and are interdigitated, because they're being recruited in similar contexts, but they do very different things. And I think a place like the prefrontal cortex, whether it's PL or IL or OFC, it's biasing and weighting with an EIGH, weighting the different outputs so that the organism now responds in a way that's tilted this way or tilted that way, and the integration of input and response outcome changes the balance, changes through processes of plasticity, changes which of those circuits gets priority in that particular context. And that's a very dynamic process. So I think these regions represent multiple circuits, and they talk to each other, but they're also sort of independent. And it really is the relative activity that is the function.
Yeah.
Right.
I think that's a good point, because I like, for even within dorsal regions, like ACC has really strong projections to dorsal striatum and less dense projections to ventral striatum, whereas right below it, PL has really dense projections to the ventral striatum, which is more in line with its role in emotional processing. Whereas ACC, I think, is more important in doing cost-benefit calculations, and then executing some motor response based on that. But yeah, I think that we don't understand very well what the interconnections are doing, because it's been difficult to study without all these precise viral tools. But now we have tools that we can start to do that.
But your work really starts to show it, I think, because you can just find these are the neurons that were engaged right now in this. And it's not a very large proportion of the total, so that's fine. That part of the cortex can do many things.
Yeah, these intersectional tools, I think, are going to reveal a lot, because in my previous study, I didn't show this, but if you just trap the cells that are active during recent or remote memory, they look exactly the same as like all of PL. But once we did this intersectional approach, we could start to really see subcategories like these TEA claustrum neurons that we would not have been able to pick up without using these logical gates. So I think that's a cool thing to keep going with.
So even in one situation, there are sort of separate microcircuits or something within the cortical region that are engaged in that.
Yeah, and I think they're just tuned up and down based on, incoming inputs, and probably interneuron. I think another area we need to focus on is understanding how interneurons are dynamically regulating microcircuits. Because, like, for instance, we have these avoidance assays that we also do. And you can see in PL, there's a big increase in activity when the animals go into a safe zone. And then once they're there, there's a huge decrease in activity because they need to go and then they need to stay. So you can imagine that that's a shutting off of competing circuits. And actually, we found that that's developmentally regulated. Adolescents and juveniles don't have that shutting off, and they go on and off out of the safe zone at much more than an adult who's just like, okay, I'm just going to wait here until threat is gone. So I think that maybe that signal comes from IL. Who knows?
I think there's a lesson in there for people who are recording neurons in behavior and are in the habit of just saying these neurons fire at this moment and these neurons fire at that moment, because the neurons change when they fire. Mm-hmm. And we know that just from looking at motor neurons. During a movement, motor neuron will speed up and slow down and speed up again. And this one movement, but the neuron has this crazy temporal profile of activity, which has to do with the dynamics of that movement.
Well, and the tools that we have now allow us to specify what neurons we can record electrically or using calcium indicators and other things. And the viral tools that we have now, which go retrograde, anterograde, you can use different promoters to prompt a differential expression. We can pick neurons of a specific phenotype that are also receiving input from a specific afferent, sending projections to a specific target, and ask, what do they do? That's different from the neuron right next to them that may have the same phenotype but different connectivity, may have the same inputs but different outputs. It really gives us powerful tools for understanding the microcircuitry.
Yeah, that sounds great. Of course, it's another challenge, because now we have this temporal profile of what the neuron is doing. But, sorry, I have failed to connect my power.
Yeah, Alan Carter has done, some of this work, right? Like pinpointing those cells in the prelimbic cortex that project to the amygdala and that receive inputs back from the amygdala, right? And they are more engaged.
And it's a small population.
Yeah.
Right? It's a small population. And I think that's how we're going to understand the complexity of the function.
Right. But that's not to say that, many of our tools still take, larger overall shots at what many of these neurons are doing during a specific situation, and that used to be a really cool thing to evaluate complex neural systems with the entire organism, right, in vivo. But now with the development of better tools, it's kind of like backfiring on us. It's like it's like very difficult to come up with just one specific answer for our manipulations.
Add to that now the ability to manipulate those same circuits and those same cells with the same precision, right, with dreads and virogenetic or the optogenetics and other mechanisms. And you're right, we have a tremendously powerful toolbox now, and the complexity is exponential.
Right.
But that's how we'll figure it out.
But the analytical toolbox hasn't kept up.
Not yet, but we've got AI now and machine learning and
I'm sure glad to hear it.
Yeah, people has begun to implement AI and computer vision analysis of to tease apart like even very fine movements, and I think Jason has been doing an amazing job in these aspects, even from primitive behaviors like leg extension as a strategy for avoidance.
So what do you think that, so if we're looking at, I hate to harp on the same thing, but if we're looking at a motor neuron, we could look at the firing profile of a motor neuron during a movement, and we could try to interpret that and make sense, and people have done that, it even works. But if we look at, for example, you saw profiles of neurons firing during the presentation of a cue, which was a couple of seconds long, and the neuron would start out firing fast and it would slow down, and maybe it would speed up again later, and there were these, and this was consistent. It wasn't just noise. This was really happening. So probably that means something.
Yeah.
You think?
I mean, they may have to do with these ideas of like you have a behavioral context because they're in this box where they know there can be cues and something bad can happen. So some may be setting like the cue context, right? Like they're in a box, and then when the cue comes on, the threat level has increased dramatically. So some of the early activity may relate to threat level, whereas some of the later activity may be predicting when the outcome is going to arrive. But I also like Jason's idea of having these little filters that you can use to sort of select. Maybe it's a way to connect the cue with the appropriate behavior in sort of a stochastic way.
Yeah, I think that idea, it's somewhat new to me, but it's been around since the early 90s as far as I know, from Tommaso Poggio and also Puget and Sinowski later. It's you got these little oscillators, and you can make essentially it's just like Fourier synthesis, or if you just have a bunch of sinusoids or varying signals, I can make that. If you just add them up in a linear way, you can make them look like anything. So I can take a bunch of these flexible neurons in PFC and make them do any transformation if I just sort of learn some linear weights, which can be done pretty easily with plasticity, I guess. So that's kind of a cool theory. But there's
a lot. Don't have to be oscillators. There's a choice of basis function.
Yeah, exactly. Whatever, any complete basis set. Yeah, exactly. Yeah. So I think that's a really cool.
So maybe
idea.
when we're looking at that, we're seeing some of that basis functions. Like if that's the case, they might fall into simple categories that we could see. Oh, there's one of those. Yeah. Is that the way it is? Or are they all over the place?
Well, I think a tricky part of studying this in PFC is that, at a given time or day or whatever, you're going to see those signals, but they don't necessarily come from the same neurons. So it's kind of hard to study them. I mean, you'd have to do some sort of closed loop process where you're identifying them in real time and then manipulating them if you really want to understand their function. But I think other areas may have a little more stability. But I sort of think of PFC as like a grab bag where there's, of course, plasticity, but it's within a population of neurons that probably can do a certain thing because of their connectivity. And in a given, I don't know how many of them will undergo some sort of plasticity after an experience, but at a given day, you get some of them participating, and then the next day, a different subset of them. And then probably the sparse coding is really important for flexibility. And
So one of the
Context might change all of that, right?
Sure.
Physiological context as
Yeah.
well as environmental context. I mean, we study stress, but there's inflammation, there's sickness, there's age, there's sleep, all of these things reset or alter the dynamic weighting of activity and input-output relationships in the prefrontal cortex.
One of the things that impressed me was that you saw early after learning that there was a sort of predominant, there was a kind of response that was highly represented,
Yeah.
and then later, different one, with the most highly represented one. And I'm wondering if, because that it isn't completely that bad, it's like
Sure.
maybe a systematic shift.
Yeah, right. But I mean, even within those, you might have those kinds of responses are predominant at that point in time. But even it might not be the exact same cells every single day. But yeah, we do see that like there's this shift from, at least in this cell population, early suppression, and we don't know if that's important for the recruitment of the population, because what we saw could actually be functional subsets within an anatomically defined class, or it could also reflect some sort of functional change within the same neuron, but we can't really distinguish those. So I think there's a lot of interesting directions to sort that out.
Most of the directions, from the general perspective of disease, human disease, right? A typical effect of chronic stress, for example, is depressing neural activity in the PFC. Right? But that doesn't mean that overall PFC in general gets depressed, because data and even fMRI scanning in PTSD patients, for example, show that different regions of the PFC turn into different beasts, right? With the more dorsal showing more overexcitation versus the more ventral showing these depressed states during mental disease, right? And perhaps, changing that, tilting the balance off in between these different regions, going back to the point raised by Jason of differences in between PL and IL, or dorsal cingulate cortex versus ventral cingulate cortex, then the question is, well, we know quite a bit, but we don't know how to manipulate, these circuits to bring them back to normal states. And I think, the work by David is showing that at least, just promoting overall cognitive functioning of, for example, the ventral PFC seems to have, very beneficial effects.
Well, and also there's a difference between activity and reactivity, right? The overall level of activity in a region versus how it responds to input and which inputs are dominant.
Right.
Those things can change. I think that I think we're finding the subtlety of pathology is really in things like that. It's not just overt overactivity or underactivity. It really is the connectivity and the pattern of activity in responding to different inputs. And I think the things that are effective therapeutically are restoring a degree, and I hate the word normalcy, but it's restoring optimal responsivity in this region. Even if the overall level has shifted, the responsivity now is brought back to where it's responding to appropriate stimuli and selecting targets that should be getting that input at that time.
Right.
We have some kind of roadmap to use what we know about PFC to do something about psychiatric illness.
I think we are very far away from it yet.
I mean, it's, the shift in thinking, again, in terms of we move to a more translational area, the shift in thinking that happened, really starting 20 years ago with revision of the DSM, there were task forces at the ACMP, and NIH eventually then adopted the RDoC, which is really speaking to the dimensional approach, dividing behavioral dimensions, right? And
RDoC.
the RDoC is a research domain criteria. And, it defines, now we're not talking about syndromes. We're talking about dimensions of behavior that you can study from the cell, the molecule, the transcription factor, through phenotypically defined cells, through brain regions, through circuits, to whole organism behavior, and ultimately to clinical conditions. And parsing out illness into these dimensions that can be ascribed vertically now to a neurobiological system allows us to approach psychiatric disorders in a much more biologically grounded fashion and to study things like that, right? And so I think that's where we're going to get the advances in therapeutics, because now we have neurobiologists who are speaking the same language to psychologists and psychiatrists and demographers and population scientists, and we're starting to understand brain function in that same way, ascribing pieces of illness to biology, and and the medial prefrontal cortex doesn't do everything. It doesn't do everything in every illness. It does a piece of illness in specific contexts, and other things are also dysregulated that combine with it. It's moved to precision medicine in psychiatry, which I think makes a lot of sense.
Maybe that's a good place to start. So positive note. I feel much better. so
Dimensionally speaking.
Very much, yeah. Dimensionally speaking, I feel better. Thank you, David and Jason, Laura, Tony, and this has been Neuroscientists Talk Shop.